Give her brilliant talk about machine learning for precision autism care. Ali.
Ready?
Ready.
Okay, so we just been talking about autism for the past hour and other quantifying developmental milestones. And I just want to remind everyone that autism is a diagnostic label that we apply to very different kids. And they are all characterized by reduced social and communication abilities and increased repetitive behaviors. But under this single label, you'll see huge variance. For example, in language, from minimally verbal to highly verbal, in cognition, from intellectual disability to gifted, in trajectories, some regress, others steadily progress. And many have shared co-occurring conditions, such as sleep and GI issues, ADHD, epilepsy, anxiety, and more. And averaging across this mix can really mask what helps who. So when we treat autism as one thing, let me see if I can use this. We average a weight signal, trials underperform, families get trial and error care, and prognosis lack patient-level specificity. And that's a big problem because it's now estimated that 1 in 31 US children gets this diagnosis. You probably all know that we currently diagnose autism based on observable behaviors using structured assessments. And that's necessary, but it tells us what we see, not why it's happening. And you also probably know that autism is highly heritable. And if you don't understand any one of the jargon that I'm using, please ask me what it means. I mean that there's a very substantial genetic component to autism risk. And so the question is, why can't we just use genomics for risk assessment and for enabling early and accurate diagnosis? And the reason is that the genomic basis of autism is very heterogeneous. Just because there are many ways to alter human brain development that might result in autism. And the more we know, the more we realize how much we still don't know. And Thomas will speak about this after lunch, so I'm not gonna steal your thunder right now. I'm just gonna let you know that still today, the cause of most cases remains unknown. And question is, what can we do to overcome this? So we can think of autism as a mixture problem and think about it as partially overlapping etiologies, causes for autism that produce similar behaviors. And if we look through only one lens, genomics or behavior or immune function, it's difficult to disentangle that mixture because each modality per se can have small and noisy effects. But when we analyze orthogonal modalities, we might be able to raise the signal-to-noise ratio. And when independent modalities all point in the same direction, we can get a mechanistic story, not just correlations. So essentially, our goal is to move from the question that we just asked ourselves, does this child have autism, to the question of which autism does this child have, and ultimately practice precision neurodevelopmental medicine. And I want to show you how we can use routinely collected EHR, electronic health record data, to define and characterize autism subtypes. And this work was done with a very talented Gal Brown, and Shirley Freilichman did all the heavy lifting, and it was done in collaboration with Eitan and Yair at Ben-Gurion University. So the data substrate, using Noah's terminology here, is this massive, let's see if I can use this now. Nope, I can't. Nope. Okay. So it's this massive data set from 69 million patients, and that were seen between 2000 and 2017 across the United States. And it includes more than 500 million encounters, more than 900 million diagnoses, and in this massive data set, we identified more than 136,000 autistic individuals according to CDC definitions. And we wanted to define autism subgroups by focusing on co-occurring conditions, or what we like to call comorbidity profiles. And the reason that we focus on that and not on symptoms and not on developmental milestones or on severity or on other traits that can be measured across different data sets is that they point to underlying biology, and they are less instrument dependent than, for example, behavioral assessments, and they're easier to replicate in independent cohorts, and they are orthogonal to the core autism measures, so we're not double counting the same things, and they're clinically interpretable and potentially actionable. Just getting into some technical details here, just so you know what we did. We focused only on those children that had a diagnosis and before 10 years old, and we cut their data at 10 years old. And the reason for that is to enrich for co-inherited comorbidities as opposed to acquired comorbidities. And after outlier removal, we were left with about 64,000 children with autism, which is quite a large number in this domain. And then we had to do some feature engineering here. So we take the diagnostic codes, ICD codes, and we map them to PheCodes, which are systematic terms that are commonly used in EHR studies to aggregate diagnostic codes. And we went even farther, and we climbed up one layer in this hierarchical diagnostic or phenotypic tree to have less features and a little lower resolution to be able to easily compare across individuals. And then what we have to work with is this binary vector that includes 63,673 rows and 457 columns of 0 and 1. And that represents the existence of a comorbidity in any one child with autism in our cohort. And then I like genomics. I do a lot of genomics, so I borrowed the methods from single-cell RNA sequencing to this domain, and we just followed the standard single-cell RNA sequencing analytic framework. But instead of genes by cells being measured, we're measuring here the existence of any one comorbidity. So what we did here is we reduced the dimensionality of the data using a common dimensionality reduction technique called PaCMAP, and then we used Hamming distance as a distance metric to reduce the dimensionality from 63,000 times 457 to a two-dimensional data. And we then clustered it using this hierarchical clustering approach, HDBSCAN. And because we like single-cell RNA sequencing, we did all the exploratory data analysis in the single-cell portal, which is a way for me to share the data with you if any one of you is interested in exploring autism subtypes and assessing any covariates or any confounders, or if you have any interest in any specific autism subtype. All in all, out of this huge, massive data set, we identified 36 clusters, and most of them exhibited significant associations with specific PheCodes, which is our phenotypic nomenclature here, and that suggests to us that we have meaningful patterns, we have detected meaningful patterns within this data. So this is a typical plot you would see in single-cell RNA-seq studies where we visualize the relations between clusters and measurements, and it's a dot plot, standard dot plot, but you also have the size of each cluster here on the right, and so the color of each dot is related to the odds ratio, to the enrichment of any one phenotype within a cluster, and the size is the significance of that association or enrichment. And so I'm just giving you a few examples. I'm not going to go into each and every subgroup. For example, this phenotype of dyslipidemia, having altered lipid levels, is significantly enriched in one single cluster, very strongly so, in cluster 14. And we have here mood disorders. We see that they are enriched in multiple clusters, not just one. And we have here symptoms involving the digestive system that are also associated with or enriched in multiple subgroups or clusters. And I'm going to go into some more details about them in a minute. I just want to let you know that this is a nice association test, but we can also characterize these subgroups using a classification scheme. And we use light GBM to classify patients into the identified clusters that we identified using the complexity reduction and clustering approach. And then we use Shapley values, which I'll explain in a minute, to understand the most predictive features of each subgroup. And we did it using multiple approaches, just using the data available at the first encounter of each patient where we have an autism diagnosis present, or taking all the data up to age 10, just to understand the information content or the predictive value of each feature of each diagnostic group for each cluster. So now you are looking at several plots, actually 3, to be precise, 3 plots of Shapley values of 3 different clusters. And I'm just going to explain one of these plots. Here we have the 10 top features or groups of diagnoses on the y-axis. On the x-axis, we have their Shapley value, which is a summary statistic of the contribution of each of these features or phenotypes it's a summary of the predictive value of each of these features to the specific subgroup. So how is epilepsy essential for defining this one subgroup? And here we see that, for example, this one feature, epilepsy, highly contributes to the notion of this subtype or subgroup being what it is. And the reason I know it is because it has positive Shapley values, as you can see here. It's also important to know the color coding scheme here, where red implies presence, 1, the value 1, and blue implies absence, the value 0. And so this one subgroup that we're looking at is characterized by presence of epilepsy, and that is the most important feature within this subgroup, and the lack of having other encephalopathies, other neurological disorders, no sleep disorders. So this is kind of like a very clean epilepsy subgroup. So these are individuals with autism and epilepsy, and nothing else, as compared to this one cluster that has an enrichment of a bunch of individuals with autism that also have comorbid sleep disorders, neurological disorders, epilepsy, and other immune issues, hearing loss, anxiety. So these Shapley values are actually a metric that we give each phenotypic label as a quantitative metric of its contribution to the classification performance here. So how important is the fact that these individuals in this cluster have a sleep disorder to the assignment of an individual to this one autism subgroup as compared to another subgroup? One final very simple example is this subgroup, individuals with autism and an asthma diagnosis, and they don't have epilepsy, no constipation, no sleep disorders, etc., no other demographic covariates that might affect our clustering or our subtyping. So you can use various approaches to characterize our subgroups. But okay, nice. We identified these 36 subgroups, and we know their very important characteristics and what's not so important. How could that be useful right now? So one way that we can make use of this data is to ask how informative is the subgroup assignment to later healthcare utilization or to follow up clinical trajectories. So out of our 63,000 individuals, about 9,500 had data after the age of 10. And we can examine how informative is a subgroup assignment to our understanding of future healthcare utilization or future additional diagnoses or any other clinical trajectory related questions you might have. How is this knowledge at age 10 of which autism subgroup a child belongs to teaches me about their future trajectory? And of course, based on this knowledge, you can ask you can come up with ways to use this data to optimize care for these individuals. So that was an overview of just having the idea of autism subgroups based on comorbidities. And now I want to just give you a few examples and show you how using other research cohorts can help us triangulate the different etiologies that make up the very heterogeneous autism spectrum. So I previously mentioned interest in dyslipidemia, and I highlighted it in our dot plot that I showed you before. And I'm just now showing you again this one cluster that is enriched with individuals with autism and type 1 diabetes. They also have hypertension, dyslipidemia. I'm showing you a forest plot that shows you the odds ratio of having any one comorbidity for individuals within this subgroup as compared to individuals outside this subgroup. And this is just based on simple logistic regression that takes into effect demographic covariates. This one cluster or subtype is, as I said, enriched with dyslipidemia, type 1 diabetes, hypertension, cardiac arrhythmias, hypothyroidism, and other cardiac issues. And these are kids less than 10 years old that also have an autism diagnosis. These are not adults. We can also ask, how are these comorbidities correlated? What is their co-occurrence in our data? How close are they within this subtype? Meaning that how many individuals that have hypertension also have type 1 diabetes? And we see here pairwise distance metrics, again using Hamming distance and the p-values that are associated with them. And the most correlated ones are lighter, and the least correlated ones, or the farthest ones, are darker. After learning from Ken that we should not get epidemiology wrong, we also want to ensure that there aren't any obvious confounders that are affecting our observations, and there aren't. I just finished showing you this one cluster associated with dyslipidemia, type 1 diabetes, no obvious demographic confounder. And I also want to show you other clusters in our data, cluster being synonymous with subtype or subgroup of autistic children that also have an enrichment with of dyslipidemia diagnoses. So we have a total of 4 clusters that have that we see kids with dyslipidemia-associated autism more than the general autism population. And one of them is a cluster of obesity, in which 20% of the kids also have comorbid dyslipidemia. Again, all of these are autistic individuals. Another one is a cluster of mood disorders that are significantly overrepresented in kids within this subgroup, and 14% of them have comorbid dyslipidemia. One last one, individuals with conduct or impulse control disorders, again, 14% with dyslipidemia. This is in comparison to a baseline dyslipidemia prevalence in the autistic population that we're currently studying of 5%. That is also higher than the general population, matched as best we can. Okay, another cluster, never mind. So, okay, now we did this very nice EHR-driven subtyping, identified some clusters with dyslipidemia, and of course we want to validate it and replicate it and understand where these observations are coming from. And I want to introduce this study that I did with Yuan Luo that was a vast integration of various multimodal data that in an unbiased way also reached the conclusion that there is a subtype or subtypes of autism that's characterized by dyslipidemia. I'm showing you an overview of this study just to let you know that here we integrated transcriptomic data with sexual dimorphism data, as Yair taught us previously. Boys develop later than girls, and you probably all know that autism is a male-dominant disorder. About 80% of the kids that receive this single diagnostic label are boys. So we wanted to focus on those genes that have a sexually dimorphic expression pattern during early brain development, which is when we think autism arises. We integrated genomic data onto that and focused on variants that segregated with autism. And we validated that all of these findings in an additional clinical cohort, actually multiple cohorts. So that really nails the idea that there are some children with autism that also have altered lipid regulation, and I'll show you what we found and how we dissected this even further. So after this very lengthy overview of this study, I want to tell you that we started with the idea that we know autism is male dominant. We want to focus on those genes and combinations of genes, and actually all of this was at the single exon level. We want to focus on those genes that are very active during prenatal brain development, which you can see here, days 0 to 256. And we like to also talk about developmental periods. In each period, other functions of the human brain develop. So the prenatal period would be period 4 to 7. So we focused on those exons that have a sexually dimorphic, so different expression between males and females in those periods of interest, prenatal period or periods 4 to 7. And for that purpose, we use this very nice Allen brain data set. It's called BrainSpan, which has massive amount of transcriptomic measurements, and took a lot of compute power to calculate everything that we were able to infer using this massive data. And in addition to transcriptomic data, we also examined whole exome sequence data, aggregated across several data sets, and following standard variant calling approaches, which I just flipped that in a minute, but just getting all of this done in an aggregated manner, that's like a year worth of work. Aggregating across different genomic studies and harmonizing everything. And basically, across these studies, we had 1,700 discordant pairs, meaning that there is one child with autism and another child that has no autism diagnosis, although he or she were assessed for that, so we're sure that they're not on the spectrum. And then we also had 50 families that had at least 2 affected SIBs. And in those families, we're looking for shared genetic variants, and in the discordant pairs, we're looking for variant that's found in one of the siblings, but not in the other. And then we focus on those variants that we think might have a deleterious effect, and we call them likely deleterious variants. So then we come up with these clusters of neurodevelopmentally co-regulated, sexually dimorphic segregating deleterious variation. So sets of exons functioning together in a sexually dimorphic manner that host in them variants that follow, that segregate with autism in the families that we studied. And among these many clusters, many of them were enriched with functions that we already know are implicated in autism, such as synaptic function, immune function, growth and transcriptomic regulation functions, all functions that we know are related to autism. But several clusters, including this 184 gene cluster, were enriched with metabolism of lipids and lipoproteins or other lipid-regulating functions. And this is this one pathway. It's a reactome pathway. The variants in this pathway were in genes that I'm showing you are sexually dimorphic in the prenatal periods, and it's a huge cluster of exons that we think functions together, and both based on its expression profile in human brains, but also based on what we know about lipid metabolism functions, for example, via this reactome pathway. So now within this framework of aggregating this massive amount of genomic and transcriptomic data and doing all these triangulations and finding this functional convergence, finally we have a hypothesis that we can test. It seems like lipid dysregulation might be a convergent etiology in autism. And I didn't show you, but part of the genes that we found to be extra or to have a higher probability of being dysfunctional in autistic individuals are this LPL, lipoprotein lipase gene, and LDLR, low-density lipoprotein receptor, the gene encoding it, of course, and other genes in this pathway. And we said, well, okay, if these genes are dysfunctional, as we predict them to be, but we don't really know, then our hypothesis is that LDL will not enter the cell and do everything that it's supposed to be doing in the cell. And if LDL is not entering the cell, then and LPL is not hydrolyzing triglycerides, then our hypothesis is that we will see LDL particles in abnormal levels in blood. We'll also see triglycerides, and we'll also see VLDL remnants, which subtype of cholesterol. And this is a forest plot showing the association of an abnormal blood lipid test with autism in these three commonly performed lab blood tests. And so we see an enrichment of abnormal blood lipid profiles in a subset of individuals with autism. And of course, you can say, well, it could be related to lifestyle, it could be related to metabolic status, it's obviously age-related, it's obviously sex-dependent. So we were able to match all of that, males, females, and metabolic status, and show that across all these strata, predefined strata, we are able to see differences in blood lipid profiles. This is for triglycerides, but I can show you similar results for other lipids. And one more thing that we can do now, looking at another data set, is to ask, what about dyslipidemia diagnoses in individuals with autism outside the EHR data set that I mentioned, the Cerner data set? Now, this is a completely different data set. Coming from healthcare claims from Aetna, a healthcare insurer in the United States. And here we had about 39,000 families with ASD, so with at least one child diagnosed with autism. And we used repeatedly matched controls, the best we could match them based on zip code, age, sex, etc. And all this work was done by Nathan Palmer. And looking at the kids with autism as compared to their unaffected siblings and matched controls from other families, we found that dyslipidemia diagnoses at large were highly associated with an autism diagnosis. So similar to what I showed you before, using the Cerner data set, this is a totally independent data set. Again, finding an enrichment of dyslipidemia diagnoses in a subset of children with autism. Altogether here in this one data set, we're talking about 6.5% of kids with autism also have a comorbid dyslipidemia diagnosis or healthcare claim that recorded that. And we can also look at the parents because we said we're looking for co-inherited conditions. Theoretically, we expect, if we're talking about very young kids with dyslipidemia diagnosis, it's most likely inherited. And when we look at the fathers here on the right, mothers on the left, we see an enrichment of dyslipidemia diagnoses in both parents, or either parent. Very significant because we have such a huge cohort. We were also asked about drug use because many kids with autism also take atypical antipsychotics to deal with other comorbid mood issues and other neuropsychiatric conditions that are often co-inherited. And we're able to show that we were able to match by drug use and show that independent of drug use, we still see abnormal blood lipid tests in individuals with autism, more than matched controls. So what am I doing on time? I have time. Okay, so obviously we're not the first to study autism, and although we're using more data-driven analyses, there's always a history of the field, and I always like to see if my studies resonate with previous observations. Luckily they do, and this finding can explain Smith-Lemli-Opitz syndrome, SLOS. This is a rare Mendelian disorder of impaired cholesterol metabolism. It's a very rare, 1 in 50,000 births, caused by a loss of function of a cholesterol metabolism gene, DHCR7. And in the syndrome, the vast majority of kids also get an autism diagnosis. And so this is kind of like our gold standard Mendelian case showing how altered cholesterol metabolism is strongly associated with autism, but it's a very rare corner case. So we can also, if we're already dealing with Mendelian genetics and single gene hits, we can also go to mice and ask what is the phenotypic overlap between mouse models of dyslipidemia in orange and mouse models of autism in blue. And the SLOS mouse with this functional DHCR7 gene is shown in red. So this would be kind of our gold standard overlap between dyslipidemia and autism. And this multidimensional analysis that I haven't shown you all the details of found that just taking a huge data frame of all phenotypes that were measured across these mice and clustering them, we see that some models of autism are more related to models of dyslipidemia than to other models of autism. Now, we already discussed how heterogeneous autism is, and it's no surprise that we have all these subgroups of autism within single gene mouse models of the disorder. So to summarize here, we see that some models have a very similar phenotype to dyslipidemia models. Okay, summary and conclusions about this one subgroup of individuals with autism that's characterized by co-inherited dyslipidemia. We have integrative work that suggests that lipid dysregulation defines specific autism subtypes. We've done this integrative genomics work to show that autism segregating deleterious mutations in sexually dimorphic genes that are also neurodevelopmental developmentally co-regulated, converge on lipid metabolism functions. We found altered blood lipid profiles in children with autism as compared to matched controls. We found enrichment of dyslipidemia diagnoses in individuals with autism and their parents as compared to matched controls. Again, enrichment of dyslipidemia diagnoses in individuals with autism as compared to their unaffected SIBs. Shared behavioral phenotypes that I just showed among mouse models of autism and dyslipidemia. And we saw in the EHR data set that I started with that in autism, among children less than 10 years old, dyslipidemia was associated with type 1 diabetes, obesity, mood and conduct disorders, and others. Okay, so that does it for my description of this one subgroup of kids with autism revealed through this EHR work, purely data-driven. Right, so I want to just talk about another example. And how much time do I have? I'm good, okay, so.
At least 23 more minutes.
Woohoo! All right, so I have this. We have this very common comorbidity in autism that has to deal with GI dysfunction. Many kids with autism also have various degrees of gastrointestinal problems. And that could be related to their eating habits, but it's in these kids that are very young, we see it very loud and clear. So it might be related, but it's also inherited. So we have a few subtypes here that are characterized by GI issues. And taking a step back, and I want to tell you about this very cool observation that started several decades ago. And what you're looking at here are fever cabinets that were very common back in the day. This is an editorial from May 1936. And the editorial says, the use of artificially produced fever has become established therapeutic procedure for the treatment of certain diseases. And in the 1980s, there were several reports, including this one in Nature, that said that report by caregivers in a neuropsychiatric facility that noticed that when autistics have a moderate fever, they invariably display dramatic, more normal behavioral patterns, including a greater desire or ability to communicate. And there are other anecdotal reports. So, of course, this is a very intriguing question. It seems like there could be a natural mechanism to modulate autistic traits, modulate human behavior. It seems like people have associated with fever, but and I hinted that I'm interested in GI dysfunction in autism. So we really wanted to know what are the most likely neurobiological features that are related to this phenomenon that has been described for so long. And for that, we used another research cohort that we're triangulating across data sets, just like Yair showed in his research. So this is a very nice and well-studied research cohort from about 2,600 families with one child with autism, including genomic data, very large and diverse behavioral questionnaires, medical histories, and among all of these massive amounts of data is a question: does your child seem to show any improvement in symptoms of autism when he or she has a fever? So now we have data to correlate with an outcome of is there a report of a child improving during fever? So this was work again with the omnipresent Eran and the very talented Efrat, and using in this data set, Efrat showed that 1 in every 6 children with autism is reported to demonstrate behavioral improvements across multiple autism domains, including cognition, which is not the most intuitive improvement you might imagine when a kid has a fever, better communication, less repetitive behaviors, more social interaction, and temper, which you might expect kids with a fever to show different tempers. At least my kids do. Here we did some data analytic techniques to just reduce the complexity of the data, but still in a more data-driven way. We clustered variables and focused on the set of variables, the set of clusters that is a priori more relevant to our outcome, which is, does a child improve when they have a fever? And so the first thing that the data shouted at us is that individuals that are on the lower end of the spectrum, meaning that they require more support, are more likely to be reported as those that improve when they have a fever. Now, is it because it's easier to see a change? Is it because they are actually the ones that improve? The jury is still out on that. We still need to dissect that. But what I'm showing you here are a bunch of behavioral metrics, including IQ, social interaction scores, verbal communication scores, and other metrics that were used that, importantly, are evaluated by different specialists, some by clinicians, some by teachers, and others reported by parents. And all of them are consistently showing a difference in the distributions of these behavioral questionnaires or assessments between kids reported to improve when they have a fever and those that were not. And more importantly, because we care about the neurobiological mechanisms that might be relevant, we found that the greatest predictors of fever response are severe GI disorders. So here, I'm starting with part B. I'm showing you the sum of reported severity of GI disorders across different disorders. And we see a very tight correlation between the total sum of reported severity and the likelihood to be reported as a kid that improved when they had a fever. We also see a very strong association between reports of maternal infection in pregnancy and chance of being reported to improve. So these were the greatest predictors. Of course, we want to know what else, not just direct predictors. So we have this very naive network showing that not only maternal infection in pregnancy and GI disorders are associated with fever response, but also inherited factors such as autoimmune disorders in the family, neuropsychiatric disorders in the family, language disorders in the family, and here on the bottom you can see the pairwise associations between any of these factors. So it suggests that the fever response might be inherited, and it also points to the involvement of pro-inflammatory cytokines in this phenomenon. And the reason that we think that pro-inflammatory cytokines are those that connect these dots is because of this very nice autism model in mice that when you inject virus-like particles, just poly-IC, never mind what it means, Michal is nodding. So you imitate a viral infection in pregnancy in a pregnant mouse, there is a much greater chance of the offspring to have some form of neurodevelopmental abnormality, as much as you believe that we can measure autism in mice or other behavioral trait. And it was shown that the gut bacteria is the missing link between immune activation in pregnancy and developmental outcomes in the offspring. And the signal was shown to be amplified by certain gut bacteria. When they were present, the signal was amplified, and the pro-inflammatory cytokines signaled to a certain brain region, S1DZ, and activated IL-17 receptors there. I'm seeing that I'm all losing you guys here. So there is a very well-defined molecular mechanism that links maternal immune activation and altered behavioral and adverse developmental outcomes in the offspring. I still have time, right? We also wanted to examine the correlates of these findings in a larger population. So now Tal Richter analyzed lymphoblastoid cell line gene expression from individuals that were part of the Simon simplex collection that I previously described. And she now asked, can we find molecular markers of the fever effects using peripheral RNA? It's not really peripheral RNA because it's a cell line that is a proxy for peripheral RNA. But still, this is the best we have. We want to understand this phenomenon. This is our only chance of modulating autistic traits naturally. And although the data isn't perfect, we're going to make the best of it. So she used this biclustering approach where we say we know how heterogeneous the data is, both in terms of gene expression and in terms of the subjects. And we are just looking for clusters within the data. We don't want to cluster everything. We want to identify a subset of patients and a subset of genes that can tell us something about the chance of being a fever responder, what so-called fever responder. So being reported to improve when they have a fever. So basically, we found that many of these clusters that were enriched with fever response with individuals that were reported to improve when febrile, genes in these biclusters had to do with immune function. We can look at single genes, but it's more informative to look at the pathway level because we know how heterogeneous autism is. It requires some aggregations of different representations to actually identify a convergent theme. So when we look at the pathway level, we found enrichment in immunoglobulin-mediated immune response, some B-cell mediated immunity. I'm going to remind everyone that these are lymphoblastoid cell lines, so we have some artifacts here, and other immunity-related pathways. So that's in line with what we saw using the clinical data only. And one more finding that was really encouraging is that when we only look at those individuals reported to have GI dysfunction, and we compare individuals reported to improve when they have a fever and those were not, again, all with at least one documented GI dysfunction, we find a significant upregulation of IL-2 receptor alpha, this is a gene that regulates the strength of the immune response. Of course, the immune response needs to be activated when needed, but also suppressed after it has done its role. And it's a very central receptor in regulating that. Other students in my lab, Tal and Shachar, were able to then follow up these transcriptomic findings and examine, again, whole exome sequence data in the IL-2 pathway, looking at variants within this pathway and examining the association of likely deleterious variants. We talked about how we're not sure about the variant impact yesterday. Just making sure I have time. And they found differential likely pathogenic mutation burden in IL-2 signaling pathways in those individuals that have that are known to have GI dysfunction, and comparing those that were reported to respond when they have a fever, and those that are not. Okay, so that's another modality that we're using to confirm the involvement of this immune function. One last thing that I'm gonna cut a little bit short is this idea of predicting using the most widely used classification framework, trying to identify those predictors of fever response in a purely data-driven fashion. We have a problem here, technical problem, that our classes are imbalanced. We have a lot more individuals reported not to improve than those that do improve, so we have this technical issue. We have to do bootstrapping and repeatedly resample individuals that will go into our predictors. And based on our resampling, we have different distributions of performance. But what you should know is that when we examine the set of good predictors, quote unquote, we find that in addition to the clinical predictors that we have previously discovered, which are the strongest predictors, such as low verbal IQ or maternal infection in pregnancy, the most informative gene predictors are genes that have a documented immune-related function. So this again strengthens the idea that immune-related genes and immune pathways are those that modulate, that can modulate autistic traits in a subset of autistic individuals. And Tal also created this nice tool that you can use and save yourself a bunch of time if you have imbalanced data and you want to classify it in a robust manner. I'm also going to mention that we also tried to avoid a bias that had to do with remembering whether your child ever improved when they have a fever. Because when you ask a parent to report, if it happened 2 days ago, they're more likely to remember that. But if it only happened 2 years ago, they're less likely to remember that. And of course, we have other biases. So we built this very nice app that we call ASD Pulse that asks parents to continuously, weekly report about their kids' behavior and separately report about their kids' health status. So there's no bias in them correlating their kids' health status with their kids' behavior. And so we had independent surveys that were answered at different times, and also took into account known covariates such as stress and other just not paying attention to the survey and just filling it up. And we were able to collect temporal data in real time and examine ourselves the relation between autistic behavior and health status. Of course, we didn't have enough kids to actually reach any statistically significant conclusion. But what we did learn is that it's so difficult to correlate these two. And there are so many variables that we cannot even imagine that affect a kid's health status and affect a kid's behavior. And just continuously collecting data is really naive. And we need to come up with other ways of doing that if we want to measure variability in autistic traits and how that relates to one's health status. And I'm going to talk about it later when we have our session, because that's something that we need to know. We need to know how variable are autistic traits. If we're going to do RCTs and we measure autistic traits at one point, how robust is it? How stable is it? So I'm putting it all together in terms of the fever-responsive autism subtype. It, we think, relates to about 15% of the autistic population, characterized by GI dysfunction, by maternal immune activation during pregnancy. These two are known to be linked via pro-inflammatory cytokines. They're also related to autoimmune disorders in the family, language disorders in the family, neuropsychiatric disorders in the family, all also have some form of studies connecting them to altered pro-inflammatory cytokines. And we found peripheral predictors of fever response in immune pathways. So again, and this summary was done by Notebook LM, which is the best way to summarize your own research. So this is another example of an autism subtype that now that we understand its characteristics and predictors and specific presentation, we can continue following up on this one subset or subgroup to better understand not only the fever effect, but also how pro-inflammatory cytokines can modulate autistic traits in a better powered way, less heterogeneous way. So my very last slide, putting it all together. We're trying to move towards precision medicine of neurodevelopmental disorders, and I've shown you how big data could potentially transform behavioral autism into biological autisms. And back to Michal's talk from yesterday, we're trying to shift from calling it cancer to calling it stage 4 melanoma with BRAF mutations and having better opportunities to provide better care. And I've shown you metabolic subtypes, immune subtypes. There are also neuro-related subtypes that we're going to hear about after lunch. And now I'm going back to Ran and Noah's talks from yesterday. As it comes to pediatric care, we still have a very long way to go to enable the practice of preventive and proactive, precise and personalized medicine. Of course, we're not even starting to talk about responsible AI. We're way back. It's really difficult to study neurodevelopment and it's really difficult to have RCTs in children. But working on it, we're not discouraged at all. And I want to thank all of my collaborators, funding, and everyone. And thanks for listening to everything that I had to say.
We do have time for a question. Who wants to go first? Please.
Yeah, so for the fever subtype, does it generally correlate with a greater severity of impairment? Do you see it all the way across?
Yeah, we see that children that require more help, they need more support, tend to be reported more as those that improve when they have a fever. But we do see it across the board. Just there is an enrichment of children on the lower end.
And is there, this is probably for later, but have you noticed a correlation with other diseases that have to do
I looked
with
into that.
dysfunction of the immune system, like degeneration or whatever?
Yeah, so I actually started looking at it just in neurodevelopment because the brain is more plastic, what I think. And I think that if it's really pro-inflammatory cytokines, there is a better chance to impact behavior. And I saw some anecdotal reports about from parents with children that have ADHD that reported that when their kid had a very severe illness, they noticed that they were doing much better in terms of the symptoms of ADHD. We also saw it with one child with schizophrenia, but these are all anecdotal reports.
Sure.
But of course it starts there, and I was happy to see that. It was reassuring, but requires follow-up.
But what about things that come from immune dysfunction that have nothing to do with neurology?
With brain function.
Yeah.
Good question, whether it changes behavior. So like IBD. If kids with IBD, when they have a fever, do they become more...
rheumatoid arthritis.
Yeah, I'm focusing on the kid side because I think it's easier to modulate the brain then. It's a really good question. We need to look into that.
Should be there, right?
Yeah, if it's real.
Yeah.
Yeah, okay, I'm on it. We have a study design.
That's all you need.
Okay.
Please.
Thank you for this rich talk and interesting. I wanted to ask if you may have any possibility to look at the microbiome of autistic versus their family members that are not autistic, because there are fairly well-known reports on animal and mice that by really controlling artificially the microbiome, you can change quite dramatically and pretty much shocking, I would say, behavioral communication, voice communication, stuff like that. And there was, at least from my recollection, 10 years ago, a big, talking about this microbiome being a kind of a
Game changer.
between the genetics, the behavior, and so on, based on those animal mice. So I wonder if it can be plugged in somehow to this very rich.
We should definitely look for microbiome data in these kids. I'm sure that it was collected because this is a compilation of single hospital data sets that were all collected into this one Simon Simplus collection. So I'm sure that some hospitals collected microbiome data because back in the day it was the thing to do. And there were reports of microbiome alterations being associated with almost any complex trait, including autism. So I'm sure that if we dig deeper, we can find kids that have that data and examine how it relates.
Families that I think they are eating the same or quite the same, but then you see probably because of immune system.
Yeah.
I mean, there is enough, I would say, clue in your studies saying that maybe their absorption, I mean, something is different fundamentally so that it will be reflected by the microbiome. Maybe just looking there will end
Inform
up
us.
being more signature
Yeah.
than these complex traits that you are trying to grab.
Dissect. Yeah. Okay, I'm on it.
Yeah, I like mice, but I think the microbiome study were coming mostly from mice and not so clear in humans. And also when they looked at the diet of the people with autism, which is very different, very specific, then if they covariate with this, the association is very low. So that. So thank you so much, because that's, I think, the future to take all this information from. I didn't get what are the input for your clustering. Is it very different what you put input and what you look at, or is it, for example, epilepsy? Did you add epilepsy in your clustering?
This is all based on EHR data, electronic health records. So we're looking at all diagnoses that a child with autism has. And about 25 of them also have a comorbid epilepsy diagnosis. Of course, there are different flavors of epilepsy, as you know.
Yeah.
And so that's where the data comes from. We have a huge cluster of epilepsy that we then subset, and we found out quantitatively that the different subclusters are better representation of the data than the one epilepsy cluster. And I showed some examples of a cluster that has only epilepsy and epilepsy with other syndromic manifestations, or epilepsy with sleep loss, the straightforward comorbidities that you would expect to have with epilepsy. There was epilepsy with asthma, and I can go over the 36 clusters with you.
So if you remove autism diagnosis, what do you see?
Just clustering all kids.
Yes.
So we've done that in a different study. And then we examined the distance between the clusters that we found in kids with autism and the clusters that we found in kids without autism. And the immune-related ones were specific to autism. So there were combinations of asthma, as I said, epilepsy, other allergies, other GI issues, that their combination was unique to autism. But it's such a massive data set, I can look at it more, and of course I might have missed something.
Yeah, and I guess autistic adults are more susceptible to get dementia, which may come from immune dysfunction in the brain, right?
Yeah.
Yes, one more.
So have you tried to characterize the different cluster for other characteristics, or specifically for autism parameters like, I don't know, age of onset or functionality measurement or other things?
Yeah, so we have a study about age of onset. And of course, again, we try to dissect the heterogeneity. So we ask, what are the different clusters that make up individuals with delayed autism diagnosis? If I have time, I can tell you about that. Yeah, but that's also a question with clinical relevance that we need to identify those that we are currently missing with our standard diagnostic approaches and potentially develop a targeted treatment strategy or a diagnosis strategy to not miss them. So we just saw how developmental time windows go by with no intervention, and we want to avoid that. Yeah, I also look into that.
So going back to the lipid metabolism, is that just a correlation, or is there some reason to think?
Yeah, so I personally don't think it's a correlation, because I showed you how it's co-inherited, how it's also found in the parents, how mouse models have similar phenotypes between autism and dyslipidemia. All of this convergent evidence suggests that it's not just a correlation. But of course, the proof will be if it will be able to modulate lipid regulation and show how that affects autistic traits.
Yeah, but how would it work?
How would we test that?
No, what's the mechanism?
Oh, okay, that's a really good question. So, the human brain is mostly made up of lipids.
Yeah.
And they make up our membranes, and they're in charge of signaling. And if you mess up the precise lipid frequencies, you have too much cholesterol, too little cholesterol, then you can imagine how it might mess up. I'm making this up, right? How it might mess up synaptic function if we mess up with the membranes or with myelin sheaths. Maybe the signal doesn't move as effectively as it should. I'm just making it up.
Yeah, okay, that makes sense.
Is there a strong association between dyslipidemia and other brain-related, any of these? So we know some myelinization-associated illnesses. Would you see anything like this? Is it known to be associated?
I haven't checked, but I will.
Maybe more to the point, Mendelian randomization. What about Mendelian randomization? Because there are Mendelian lipid disorders, and then you'd expect that if it's causal, you would see a higher incidence of autism among those affected. You know of anything?
Yeah, so presumably we did not a Mendelian randomization study, but just an autism segregation study, and we found an enrichment of likely deleterious variants in that segregate with autism in families, which is sort of a Mendelian randomization, but not really. It's like old school.
Yeah, that's
like old school.
Loss, right? The SLOS.
Yeah, that's a proof.
There's a specific example.
Yeah, right.
Found of one that does this.
Yeah, that's one gold standard example.
So we will continue this over break, and I would like us all to give a great hand to
Thanks.
And everybody is invited for another recess, and we will reconvene. Which one is it? The longer one?
An hour and a half.
It's an hour and a half, right?
Yeah.
So now.